Assalamu alaikum to all participants. I'm Anita Kesar from the Department of Zoology, Common College University, Faisalabad, on the behalf of the 3rd International Conference on Applied Zoology 2020, which is basically organized by the Applied Zoological Society of Pakistan and with the collaboration of Quaid-e-Azam University, Islamabad, Pakistan. I'm the moderator of this special session about COVID-19. In this session, we will include the talks about ongoing pandemic, which has affected us all in one way or another. So the first presenter of the session is Kaplan Dilare Noor from the Department of Nutrition and Dietetics, Faculty of Health Science, Turkish University, Edirne, Turkey. She will talk about the effect of COVID-19 pandemic on nutritional habits and approach of immune boosting nutrition. Please, Ms. Kaplan Dilare Noor from Turkey.
Hello, my name is Dilara Nur Kaplan. I'm a master's degree student on nutritional sciences at Trakya University. Today, I'm going to present you the effect of the COVID-19 pandemic on nutritional habits and approach of immune boosting nutrition. And while I'm presenting this topic, I'll mention what COVID-19 is, how COVID-19 affected our world, its effects on human psychology and life, and nutritional approach. Different types of coronaviruses have been around for so many years, and COVID-19 is one of them. It occurs as a large ribonucleic acid virus that infects the human respiratory tract. Infection can cause fever, dry cough, difficulty in breathing, fatigue, lung infection, and death. It's known to infect human airways from the luminal side, and also viruses are released from the same side, so it makes it easier to spread through coughing and sneezing. And periods of isolation can elevate glucocorticoids such as cortisol, and can inhibit many critical functions of our immune system. Of course, it's not just the periods of isolation, but the fear of contagion, quarantine, and stigma, as well as with potential misinformation overload. They both can lead to psychological stress and anxiety. And we know quarantine induces stress, depression, and anxiety, which leads to sleep disorders, unhealthy diet, and reduced physical activity. Many people's response is not to avoid food when they're stressed, but they seek out and consume energy-dense foods, especially foods high in sugar and fat. And this desire to consume that kind of specific foods defined as food craving. And when people craving for carbohydrates, it encourages the production of serotonin, which has a positive effect on mood. A strong immune response is key factor against infection and against the novel coronavirus disease. To keep immune system strong, we have to consume high amount of minerals, antioxidants, and vitamins in a healthy and balanced diet. Especially vitamin A, C, E, D, and minerals such as zinc, copper, iron, selenium are really important to strengthen the immune system. Consumption of foods that contain or stimulate the synthesis of serotonin and melatonin, such as almonds, bananas, cherries, and oats, can have positive effects on both mood and sleep. And milk and dairy products also suggest to synthesis of serotonin and melatonin because they are the main sources of the amino acid of tryptophan, and they can cause sleep. Also, yogurt is an important source of probiotics, and these bacteria exert effects on immune modulation by increasing macrophage and increasing natural killer activity and numbers. Vitamin D insufficiency is linked with advanced stage obesity, male sex, hypertension, and all of which are associated with poorer outcomes. They strongly link to a range of COVID-19 risk factors. And it has been shown that vitamin D is associated with increased anti-inflammatory and decreased pro-inflammatory cytokines in older adults. So we believe consuming vitamin D is important to help during cytokine storm relevant to COVID-19 patients with ARDS, which means acute respiratory distress syndrome. And main source is sunlight, and we can also found in oily fish, liver, eggs, fortified food spreads, and some breakfast cereals and mushrooms. Vitamin C supplementation is known to support respiratory defense mechanisms, preventing viral infections and reducing their duration and severity, as well as having antihistamine properties, which can improve flu-like symptoms. So vitamin C decreases the levels of pro-inflammatory cytokines, including TNF-alpha, and increases anti-inflammatory cytokines. Thus, it can help prevention and support of immune responses while cytokine storm during COVID-19 infection. And some of the sources are orange, red and green peppers, strawberries, broccoli, potatoes, and tomato. Vitamin E is the most important antioxidant in biological membranes of all cells. It has been investigated as a private measure for many human conditions, including heart disease and cancer. And adequate intake of vitamin E-rich diet can help patients with COVID-19. And some of the sources are vegetable oils, nuts and seeds, wet germ, sunflower oil, peanuts, eggs, tuna, and salmon. Omega-3 PUFA, it exerts a strong immunomodulatory effect by suppressing pro-inflammatory cytokine production, and macrophage-mediated cytotics and neutrophil monocyte chemotaxis. It can affect with different mechanisms. And many seeds, vegetable oils, and nuts and fish are important dietary sources. And zinc as a cofactor, it's an integral component of more than 300 enzymes, which exerts the human immune system. And the doses of more than 75 milligram has been suggested to have promising antiviral effects against common cold viruses, including influenza virus. And mild adverse effects of zinc supplementation have been reported with dosages above 200 milligram. So relatively lower dosages may also reduce the severity of COVID-19. And some of the good sources are meat, cheese, some grains and seeds, cereals, and seeded or whole grain bread. Yeah. And those are my references. Thank you so much for your time.
Thank you so much, Ms. Kaplan, for your valuable talk. Our next presenter of this session is Meena Ahmadi from Iranian Fisheries Science Research Institute, Natural Resource Research and Education Center, Kerman, Iran. She will talk about the role of fish in the treatment of COVID-19 patients. Please, Ms. Meena Ahmadi from Iran.
In the name of God. Thanks to the organizers of this Congress. My name is Meena Ahmadi. I'm a researcher at National Fish Processing Technology Research Center of Iran. The title of my presentation is The Role of Fish in the Treatment of COVID-19 Patients. Introduction. They suppress and ultimately stifle the spirit of the global COVID-19 pandemic. The world scientific and medical communities are turning to treatments containing omega-3 fatty acid and fish oil for help. Clinical trials are underway by various pharmaceutical companies to test the efficacy of a new drug based on eicosapentaenoic acid in fish oil on symptomatic patients with inflammatory cytokines. Eicosapentaenoic acid as a free fatty acid is very promptly absorbed and incorporated into the body's phospholipid membranes and then starts to act very quickly. This formula to not only modify to COVID-19 disease process, reducing harmful excess inflammatory responses, but also to do without suppressing the immune response to the virus, which are vital to seroconversion, giving the patient ongoing protection against continued viral challenge. The goal is to minimize the symptoms of infection to reduce the risk of coronavirus complications. Eicosapentaenoic acid aspire. Clinical trials will soon be run to test the effectiveness of its new drug, EPA Aspire, on patients symptomatic with inflammatory cytokines, which is believed to play a part in COVID-19's progression. A candidate drug for the treatment of patients with new coronavirus symptoms whose clinical trial has begun is based on eicosapentaenoic acid in fish oil, consisting of a novel formulation of a highly purified eicosapentaenoic acid-free fatty acid. EPA Aspire comes in gastro-resistant capsules. EPA Aspire is a new formulation of highly pure eicosapentaenoic acid in refractory capsules that is delivered to the intestine at the desired pH to ensure maximum absorption. The fish oil used in this formula is produced from pelagic species. This unique preparation has a potential to not only modify the COVID-19 disease process, reducing harmful excess inflammatory responses, but to do so without suppressing the immune response to the virus, which are vital to seroconversion, giving the patient ongoing protection against continued viral challenge. EPA Aspire will be able to reduce the risk of coronavirus complications progressing to serious outcomes like ARDS, the need for artificial ventilation and intensive care. Objective to minimize the symptoms of infection to reduce the risk of coronavirus complications. Note, prevention or treatments is not the goal. Mechanism of action EPA. EPA as a free fatty acid is very broadly absorbed and incorporated into the body's phospholipid membranes and then starts to act very quickly. Once incorporated into cell membranes, icosapentaenoic free fatty acid significantly affects the production of pro-inflammatory mediators such as IL-6 and leukotriene B4 that play a crucial role in starting and maintaining the inflammatory process in the lungs. Moreover, icosapentaenoic free fatty acid metabolism generates pro-resolving mediators and bioactive metabolism that enhance innate microbial killing and organ protection. The investigators said. Clinical trials. Participants in their trials will be eligible patients with positive COVID-19 test results. After enrollment test, patients are given EPA Aspire overnight in hospital and continue for 2 months with a 2-week follow-up. Patients are monitored for more serious consequences and inflammatory markers with the goal the EPA Aspire can reduce the risk of this in severely ill patients. The Italian researchers who developed the protocol believe the EPA FFA can reverse the course of coronavirus infections by regulating the immune response and protecting patient from its most severe side effects. A scientific review of 12 clinical trials involving 1,280 patients showed that omega-3 fatty acid supplementation could improve lung function and reduce the need for ventilation in patients with acute respiratory syndrome. Calculation. Research shows that the anti-inflammatory and antimicrobial effects of omega-3 fatty acid can help fight some infections. With the spread of coronavirus, their potential ability to help fight infection and improve lung function has received special attention. For example, a study of 642 people with asthma found that higher levels of icosapentaenoic acid and docosahexaenoic acid were associated with a reduced risk of airway hyperactivity. Another scientific study of 7 clinical trials in more than 2,000 children showed that omega-3 fatty acid supplementation during pregnancy can reduce the incidence of wheezing and asthma in children. Omega-3 fatty acid can improve lung function primarily by reducing inflammation in the airway and potentially helping to fight some infections. But there is no evidence that it can cure or prevent it. Omega-3 fatty acid can be affected by several mechanisms of inflammation. One, decreased genetic status of inflammatory compounds. Two, change in the concentration of proteins and hormones that affect inflammation. 3. Helps produce some protein with antimicrobial, antiviral, and anti-inflammatory properties. The best source of eicosapentaenoic acid and docosahexaenoic acid, fish oil, are at salmon, herring, sardine, mackerel. Here is the resource. Thank you for attention.
Thank you very much, Miss Meena. That was very informative talk. Now, our next presenter of this session is Dr. Ghulam Mustafa from the Government College University, Faisalabad. He will present the topic Computer-Aided Predictions and Identification of Phytochemicals as Potential Drug Candidates Against Middle East Respiratory Syndrome related to coronavirus. Please, Dr. Ghulam Mustafa from Pakistan.
Hello everyone, I'm Dr. Ghulam Mustafa. I'm working as an assistant professor in Department of Biochemistry, Government College University, Faisalabad. The topic I'm going to present today in third international conference on applied zoology is computer-aided prediction and identification of phytochemicals as potential drug candidates against MERS coronavirus. First, what is MERS? MERS stands for Middle East Respiratory Syndrome coronavirus. MERS coronavirus was first reported in Saudi Arabia in 2012. Then MERS coronavirus caused an outbreak of hospital-associated human infections in South Korea in 2015 with a fatality rate of 20.4%. Seroepidemiologic and virologic research revealed MERS coronavirus infection in dromedary camels with isolated viruses from dromedaries competent in infecting the human respiratory tract. Approximately 35% of reported patients with MERS coronavirus infection were died. The exact origin of all primary human MERS coronavirus infections remains unknown, but the phylogeny has showed closer relationships of human-derived coronavirus with those of bat coronaviruses. Therefore, MERS coronavirus is a pathogen of zoonotic reservoir. Talking about genome structure of MERS coronavirus is a large single-strand positive sense RNA virus. The genome size is 30 to 31 kb and encodes a large number of proteins. The functional receptor of MERS coronavirus is dipeptidyl peptidase 4, DPP4, and its expression induces the infection in humans. The MERS coronavirus genome encodes 4 structural proteins: spike S protein, envelope E protein, membrane M protein, and nucleocapsid N protein. It also encodes 16 non-structural proteins, NSP1 to NSP16, and it also encodes 5 accessory proteins, ORF3, ORF4A, ORF4B, ORF5, and ORF8B. These are the aims and objectives of this study. The main objective of the study was computational screening of phytochemicals to reveal their antiviral activities against NSP13 of MERS coronavirus. NSP13 carries NTPase activity and RNA duplex unwinding activity. Drug scanning and of best selected phytochemicals was the second objective. And the next objective of this study was admin profiling of best selected phytochemicals. This is the layout of materials methods which was followed in this study. About 200 different phytochemicals and their chemical structures were retrieved from different databases, including PubChem. Then ligand database was prepared. The 3D structure of receptor protein was retrieved and refined, and then molecular docking was done, and then drug scanning and admin profiling were accomplished. This figure is showing the interactions of top 10 ligands with NSP13 of MERS coronavirus as target protein, And then this figure is showing the binding patterns of top 10 ligands with NSP13 of MERS coronavirus as target protein. The table is showing the interactions of best selected phytochemicals with NSP13 of MERS coronavirus. Most of the phytochemicals have interactions with arginine 339. Some of the phytochemicals have interaction with arginine 139. Arginine, which is a basic amino acid, is acting as a side chain donor in these interactions. And the phytochemicals were selected on the basis of their best S score and RMSD values. This table is showing the evolution of best selected phytochemicals through Lipinski's rule of 5. Most of the phytochemicals have obeyed all 5 rules given by Lipinski, although some of the phytochemicals haven't followed all 5 Lipinski's rules. Then this table is showing the Edmond profiling of best selected phytochemicals. So this is the conclusion. Present study discovered 10 strongly interacting phytochemicals. And on the basis of chemical interaction and drug scan of these molecules, it was found that they inhibit MERS coronavirus replication. Most of the phytochemicals shortlisted in this study fulfilled some of the Lipinski's rule of 5, while fluvietolol and cubicinol A fulfilled all the criteria of Lipinski's rule of 5 and Edmond profiling as well. The results of this study will be useful for the drug designing against MERS coronavirus. And finally, these are the preventive measures given by WHO. And these are consult a health worker if you have a fever 38 degrees or higher and cough or difficulty in breathing, and inform them of your recent travel history. And then avoid close contact with people if you are sick. Then wash your hands regularly with soap and water and maintain good personal hygiene. And finally, cover your mouth and nose with a tissue or your sleeve when coughing or sneezing. These are the references which are used in this study. So, thank you.
Thank you so much, Dr. Ghulam Mustafa, for such an informative talk. Now, our next presenter of this session is Dr. Leila Azdanpe from the Animal Science Research Department, Agricultural and Natural Resources Research and Education Center, Kerman, Iran. She will talk about the effect of aromatherapy on COVID-19 patients. Please, Dr. Leila Azdanpe from Iran.
In the name of God, I like to say hello and thanks the conference staff. I'm Lali Azdanpanah Goharizi and my colleague is Dr. Jalil Zori Zahra. I'm from Animal Science Research Department, Agricultural and Natural Resources Research and Education Center, RAO, Kerman, Iran. My presentation is about the effects of aromatherapy on patients with coronavirus. What is COVID-19? COVID-19 is a member of coronavirus family that's spreading the world and due to lack of drugs at present. COVID-19 affects different people in different ways. Most infected people will develop mild to moderate illness and recover without hospitalization. In COVID-19, most common symptoms are fever, dry cough, tiredness, and less common symptoms are aches and pains, sore throat, and headache, loss of taste or smell, a rash on skin, or discoloration of fingers or toes. COVID-19 is caused by severe acute respiratory syndrome, coronavirus 2, SARS-CoV-2, which is said to be of zoonotic origin. COVID-19 is caused by a virus, not by bacteria. The most potent and effective administration method is oral administration, in which the components of the essential oil reach the bloodstream. Both methods, respiratory, also are effective. Essential oils, or EO, from aromatic plants have the main active molecules that have shown promise as antiviral agents against several pathogenic viruses. Medicinal and aromatic plants, especially those frequently used in traditional medicine, play a very important role as they contain bioactive components which could be used to develop new formulas, medicines to relieve patients with symptoms of COVID-19 with no or minimal adverse effects. Essential oils have long been known to have anti-inflammatory, immunomodulatory, bronchodilatory, and antiviral properties and are being proposed to have activity against SARS-CoV-2 virus. There are a wide range of essential oils and their components have been clinically proven to possess antiviral properties. However, the effects of plant essential oils on human coronaviruses are yet to be explored. In particular, their effects on host cell receptors have barely been investigated. How to inhale essential oils? Essential oils can be breathed in using the following methods. One method, inhale directly from a bottle held about 6 centimeters under nose. One deep breath per nostril. Do not allow the neat oil to come into contact with your skin and be especially careful about your eyes. And 1 drop of essential oil onto the palm of one hand. Bring both hands together, then cup them gently over your nose. Breathe in slowly to the count of 3 and then exhale slowly to the count of 6. And 3 method, drop 3 drops of essential oil into bowl of hot water and inhale the steam. And 4, burn 3 to 6 drops of essential oil in a vaporizer or light bulbs. Ring in order to disperse them into the atmosphere. When they are inhaled through the nostrils, essential oil molecules are taken up deep into the brain. Just how the tiny aromatic molecules pass into the bloodstream is not entirely understood as yet, but research has shown that they come into contact with the part of the brain which regulates the nervous and hormonal systems in our bodies. A study by Jack Wood and another researcher found that treatment with a mixture of oleoresins and essential oils from botanicals decreased the severity of clinical signs and lesions in chicken that carried the avian infections bronchitis virus. In the pandemic COVID-19, the time is advised to use frequently as a species and in some foods as a preventive measure to decrease to viral load of coronavirus. And the research study has shown the effectiveness of cinnamon with other ingredients to exert a variety of antiviral effects on beta coronaviruses and SARS-CoV by inhibiting of cell entry and infections, inhibiting of replication and inhibiting the viral protease to improve the antiviral immune response and reduce the formation of virulent quasi-species. These components may be effective in decreasing and preventing the contamination risk as well as treating some symptoms of COVID-19. And results show aromatherapy is a practice of using essential oils for therapeutic benefit. Aromatherapy has been used for centuries. When inhaled, the second molecules in essential oil travel from the olfactory nerves directly to the brain and especially impact the amygdala, the emotional center of the brain. Essential oils can also be absorbed by the skin. A massage therapist might add a drop or two of wintergreen to oil to help relax tight muscles during a rub down. Essential oils can make a positive impact on your health and well-being as long as you use them in a safe ways. Thanks you for your attention.
Thank you so much, Dr. Laila, for such an amazing information about the COVID-19. Now, our next presenter of this session is Ms. Maide Talabi from the Iranian Fisheries Research Institute, Agricultural Research, Education and Extension Organization, Tehran, Iran. She will talk about the influences of COVID-19 on marine mammals. Please, Ms. Maide Talabi from Iran.
Hello everyone, hopefully you are in good health. My name is Maide Talabi from Department of Natural Resources, Fishery Group in Iran. Our title presentation is about the influences of COVID-19 on marine mammals. For introduction, as you know, in the current century, the world is faced with unprecedented pandemic not only on humans but also on fish, especially on marine mammals. However, emerging viruses are a major public health problem. Approximately 75% of emerging diseases have animal resources. COVID-19 is an infectious disease caused by severe acute respiratory syndrome, coronavirus 2. The current pandemic, it means COVID-19, is related to family Coronaviridae and order nidovirales. This is divided into 4 agenda: alpha, beta, gamma, and delta coronavirus. Gamma coronaviruses. The first virus was reported in 2008 in beluga whale. A whale died from acute liver failure after suffering a short generalized pulmonary disease. The virus was most closely related to avian coronaviruses. The second one was reported in 2014 in bottlenose dolphin. The virus didn't induce any clinical signs or symptoms, indicating an asymptomatic or mild infection. Sano and his colleagues in 1988 isolated a coronavirus from a common carp that died after suffering an acute infection. Clinical signs included erythema on the appendum or hepatic and renal necrosis. The virus is transmitted to carp through the water at the temperature of 20. The pathogen was isolated from the kidney, liver, and spleen. In this slide, I can say about how wastewater introduced COVID-19 on marine mammals. Through urban and agricultural runoff or in wastewater effluent. Indeed, emerging reports at the time of publication indicate the presence of SARS-CoV-2 in river water receiving untreated human soil. The virus is transmitted to carp through the water at the temperature of 20. The pathogen was isolated from the kidney, liver, and spleen. In this slide, I can say about how wastewater introduced COVID-19 on marine mammals. Through urban and agricultural runoff or in wastewater effluent. Indeed, emerging reports at the time of publication indicate the presence of SARS-CoV-2 in river water receiving untreated human soil. The viruses contained in this wastewater and sludge is thus deposited on crops and on the soil where they are likely to survive. Marine mammals are vulnerable to the influx of pharmaceuticals, personal care products, detergents, and nanoparticles from sewage. Another invisible threat carried in the sewage to marine life is the potential influx of foreign microorganisms found in human excrement. And this figure is shown about how wastewater transmitted the coronavirus to marine mammals and is an emerging concern. Disposal materials. As all we know that plastic pollution in the marine environment is a global issue. However, this virus can survive on surgical masks and other plastic materials for several days. This protective covering as marine debris can introduce this COVID-19 from human to aquatic mammals. The sewage, especially in developing countries, directly go to the ocean without treatment and can transmit the virus to the ocean. However, shops and restaurants reverting to single-use disposable plastic items and supermarkets refusing reusable shopping bags for a period. Cetaceans establish herd and migrate to long distance beyond the political and geographical boundaries. These characteristics can increase the chances of an outbreak if a highly infectious SARS-CoV-2 is introduced in their communities. Changes in citizen science data collection. This related to reduction of social interaction for volunteers, as well as the loss of important data such as distribution and movement of marine species. However, lockdowns have also been accompanied by an increase in wildlife sightings in and around cities, such as sightings of humpback whales in the waters around metropolitan Vancouver, Canada. Whether this is a result of the overall slowdown in our lives, providing the opportunity to look more closely, or because there is less human activity allowing animals to enter spaces they previously avoided, is unclear. As can be clearly seen that the bar graph has shown about the total number of reported sightings between 2 years, 2019 and 2020. Marine traffic. Reducing the capacity rescue of marine mammals. This virus pandemic impacted the ability to observe, report, and respond to entangled pinnipeds. The majority of patients are seal and sea lion pups that are abandoned on beaches. These rescues require members to collect the pups. However, entanglements of seals and sea lions are also responded to. In addition, reducing the capacity to respond to entangled marine mammals. And finally, there are fewer boats out on the water. For example, commercial fishermen or tour operators, means that the number of injured and entangled marine mammals has likely been underreported. For discussion, as you know, coronavirus is the most important viruses that cause disease in both humans and marine mammals, and is known as new emergent viruses and has genetic mutations. This virus is a pandemic disease now, has become a systemic disease in humans, and is now considered as a syndrome. There are different coronaviruses have serologically cross reaction with each other. In addition to the clinical symptoms of coronaviruses in marine mammals are similar to those seen in COVID-19 patients. For cetaceans, quieter waters likely support better communication and hunting, but by contrast, reduced staffing of rescue crews have left distressed animals without aid and likely unable to survive. A reduction in the number of cetacean sighting reports means less data are available for real-time avoidance of ship strikes and other disturbance via the WRAS. Transmission from humans to marine mammals has already been observed. Moreover, pinpointing locations of sewage overflows and pipe leakage will allow for us to better assess where there is the most potential for wildlife to be affected. Last but not least, a consequence of poor wastewater management is the discharge of untreated wastewater carrying infectious coronavirus into natural water systems that are home to marine mammals. Thank you for your attention and taking your time.
Thank you so much, Ms. Maide, for your valuable talk about COVID-19. That was our last but not the least talk. Our session is going to be end. And now our next session about the drug discovery, biotechnology, bioinformatics, and nanotechnology will be moderated by Mr. Mudassir Hassan.